right, hello everybody. Welcome back. Today I want to build some viruses with you. So that when you look at all these images of viruses, you'll understand how they are put together. So, structure of viruses. Okay, so here are some drawings of virus particles, right? They're all different. And what can we make about the function of these particles? We have some that are enveloped, some that are not enveloped. They all look different. What do these proteins do? Are they there to impress us because they're cool looking? And actually not. They have functions, of course. Main function to protect the genome. Inside these particles is DNA or RNA, and it can't be floating around in the environment. Although in the pre-cellular world, apparently it was, but I guess that wasn't very efficient. Nowadays, virus particles are, well, that's one of the definitions of a virus, that it has a shell of some kind. So these shells protect the genome. So the proteins in the membranes make up a stable shell. They also have to recognize viral nucleic acid, not cell nucleic acid. Most of the time, there's no cellular nucleic acid in these particles. They say most of the time because there's always exceptions. You know, I have a clue for you on exams. If I ever say always, you know the answer is wrong, okay? Because nothing is ever always. These proteins help specifically encapsulate viral genomes and not cellular genomes, and we'll talk about that later when we talk about assembly. And then these envelopes that are present on some virus particles, or membranes, they have to get there by interacting with viral proteins, as you'll see later. Okay, so that is protection of the genome. Then we have to deliver the genome. The virus is going to get into the cell, which we will talk about on Wednesday. And the particle has to help put the genome in the right place. For different viruses, the genome has to go to different places. Sometimes it's the cytoplasm, sometimes it's the nucleus. And so the proteins do that. These proteins participate in what we call uncoding, that is, release of the genome from the particle. And also, if there's a membrane present, the viral proteins facilitate fusion with the cell membrane, and that's a way that the nucleic acids get out, and then transporting the genome to the right place in the cell. So a lot of functions of these proteins. Before we move any further, we need to define some terms, because there are some here that are very specific for viruses. So first, a subunit. When I say subunit, I mean a single folded polypeptide chain. A subunit would be the same as a single protein. So here we have some viral structural proteins. They're called VP, virion protein 1, 2, 3. That would be a subunit. The next level up is a structural unit, which you will also see called protomers or asymmetric units. I'll just call them structural units. Now that's the unit from which the capsids are built. And so the subunits make up the structural unit. You can see here there are 3 subunits making up this structural unit, which is outlined in black. So this structure, that's a virus particle, of course, which we call a capsid, and we're going to define in a moment. But the structural unit is what we use to build the capsid. Obviously, there's a hierarchy of things going on here. The subunits build the structural units, and the structural units build the capsid. The capsid is the protein shell around the genome. It's just protein, no membrane. Capsa is Latin for box. And that's a capsid in the upper right. And sometimes capsids are naked. Sometimes they have a membrane or an envelope around them. And that's shown on the lower right here. Here is a herpes virus where it has a capsid. You can see there outlined where the DNA is. And then around the capsid is a membrane. Sometimes the nucleic acid is not in a capsid, as we show in the upper right here. It's what we call a nucleocapsid, which is the last definition here. This is very tricky. I'm going to warn you now, and 25% of you will still get it wrong. I don't know what to do. I've been doing this for 15 years. Maybe I'm not explaining it right. Actually, I have a new slide this year, which I think explains it better. So the nucleocapsid is the nucleic acid protein assembly within the particle. It has to be a discrete substructure. So here on the left, this is influenza viruses. These RNA protein complexes are nucleocapsids because they are a substructure within the membrane, within the envelope of the particle. For herpes virus, this is a nucleocapsid because it's a substructure within the envelope. On the top there, it's not a nucleocapsid, it's not a substructure. We'll go into this a little more. Have a slide. Had a brilliant idea to make a new slide. Yes?
2 questions. So is it always enveloped viruses that have the nucleocapsid?
No, others can have too. Non-enveloped viruses can have as well.
If they have like a secondary shell?
Well, I have a slide, so let's wait for that. Aha! So why is the asymmetric unit different from that equilateral triangle? Because the proteins assemble to form that structure, not the one in the triangle. If you look at the colors, they're flipped. And that's why it's an asymmetric unit that builds the particle, not the triangle. Okay, and then finally we have the virion, or the infectious virus particle. Virion is a special word that we use to signify the infectious particle. Because when you say virus particle, you might mean non-infectious particles as well. All right, so those are the terms you should be familiar with. All right, now, size. Let's talk about size here. We did a little bit in lecture 1. So we use nanometer a lot to talk about size, and outside of this campus, nobody knows what a nanometer is. I've said to many people nanometers, they have no clue. I don't know why, but it's 10 to the 9th meters, or 10 angstroms, or 0.001 microns. Those are all the same. We use angstroms a lot. Not really microns all that much, not so useful. So nanometers we're going to talk about in angstroms. So for example, an alpha helix is a nanometer in diameter. That's the alpha helix of a protein there on the upper right. It's about a nanometer in diameter. And DNA is 2 nanometers in diameter. Ribosomes are 20. Poliovirus is 30. And Pandoravirus is 1,000 nanometers, roughly. Okay, so that gives you a sense of scale and the numbers that we're going to use when I talk about nanometers. That's what I mean. Now, a key point here is that virus particles are what we call metastable. All that means is that they have 2 states. They have a stable state where they have to protect the genome as it goes from cell to cell or host to host. But they have to come apart at some point to release the genome, right? They cannot be stable forever. So there have to be some conditions where the stability is lost and the particle comes apart. So that's why we call them metastable. And here's an example where the virus particle is attaching to a cell. It's attaching to a receptor, which we will talk about next time. And then the receptor is catalyzing a change in the particle so that the genome can come out. So there are many signals that can cause instability of virus particles, and one of them is the receptor, and we'll talk about more next time. All right, metastable particle. So what does this mean energetically? So if we look at a virus particle on its way to becoming unstable, you get this representative graph here. So this is free energy on the y-axis, just the energy built into the particle. The particles have a certain amount of energy built into them. And to come apart, they have to be in the minimum energy state. They have to lose a lot of that energy. This is why we say virus particles have not attained their minimum free energy conformation. That's what that lower part of the curve is. They simply haven't, when they lose their energy, they come apart. And so the way you prevent that is to have a barrier in between the high and the low energy state. So there's a high energy barrier here that has to be surmounted in order for the particle to become unstable. And that barrier can be surmounted by the receptor, as I just showed you in the previous slide. During assembly of particles, we put energy into the virus particles. We call them spring-loaded as a consequence. You can imagine a spring being compacted into a virus particle, and then it's released, and energy that is released during disassembly, that energy that's released during uncoding is used for disassembly of the particle, but only if the cell provides the right signal to get the particle over that unfavorable energy barrier. So this may seem a little obscure to you, but in reality, all it means is that virus particles are very stable. To become unstable so that they will release the genome, they have to travel over a high energy barrier, and that barrier can be overcome by signals that the cell provides for the virus particle. And when we talk about uncoding next time, we'll see how that works. Yes?
So does this diagram imply that like the virus particles are more stable when they're like broken apart than when they're together?
No, the opposite. It implies they're more stable when they have energy, high energy level. And then when they lose their energy, then they fall apart, right? It's like a disordered situation. So how do we get metastable particles? This is going to become more obvious as we talk about the structure today, but obviously there has to be a stable and an unstable structure, right? So the stable structure is created by having symmetrical arrangement of many identical proteins. As you will see when we talk about how these viruses are constructed, we take one or a few proteins, we repeat them many times, and that gives you a very stable structure. You have lots of similar interactions between all of the different proteins. They have maximal contact as a consequence. The structures are unstable because first, they are not covalently bonded. The proteins that make up the particles are rarely, that's why I have usually in there, right? Not usually. If I say in an exam, are always covalently bonded? No, because there are some exceptions where they are not covalent. Sorry, I said that backwards. If I ever say in an exam, are there any virus particles are never covalently bonded, then that's wrong because there are a few examples. But for the vast majority, they are not permanently bonded together. That way they can come apart very easily. And so if they're not covalently bound, they can be taken apart to release the genome, as we've been saying. So stable structure is in the way the particles are built, as we'll see today. Unstable, it's not covalently bounded together. All right, let's see if I explain that properly. The first question is, viral capsids are metastable because A, they must protect the viral genome when the particle is outside of the cell. B, they must come apart and release the genome into a cell. C, they have not obtained a minimum free energy conformation. D, they are spring-loaded. E, all of the above. All of the above. That's why viruses are metastable, because they have to be stable to protect the genome outside the cell. So that's one part of metastability. They have to come apart and release the genome. That's the other part of metastable. They've not obtained a minimum free energy conformation, and they are spring-loaded. All of that is metastability for sure. Now, when we talk about structures, you have to solve structures of virus particles. They're not automatically presented to you. And the tools of structural biology include electron microscopy, X-ray, so you can look at particles, take pictures of them, X-ray crystallography, and cryo-electron microscopy and cryo-electron tomography, and nuclear magnetic resonance spectroscopy. In chapter 4 of the book, if you want to go into these, you can read all about it. I'm not going to explain them very much. That is an electron microscope used for cryo-EM. I visited Cornell up in Ithaca a couple of years ago, and they brought me to see it, and you can see there, it's an amazing contraption. Millions of dollars these things cost to shoot an electron beam through a specimen. Electrons have very high resolution, much more than light. Light is too big, you can't see a lot. So let's explore this a little bit. The beginning of structural virology, where we started to see that viruses have a structure, was in 1940, when Helmuth Ruska, he invented the electron microscope and used it, he invented it a few years before, and he used it to take pictures of viruses, which, remember, up to that point were thought to be liquids. And he saw that they were particulate. This is an E. coli with a virus attached to it, a bacteriophage, and you can see that it is particulate. And that is the original paper where he published this. So from that point on, we can see virus particles, we can see what they look like, and we can compare them. Now, in order to see a virus particle under an electron microscope, you have to stain it, because biological materials, as you may know, don't have a lot of inherent contrast. And so you have to somehow make contrast. One way is to stain it. There is another way, which we'll see in a moment. But for electron microscopy, you can't use like a stain you would use for light microscopy, because the electrons will go through everything. It won't hold anything back. What you do use for electron microscopy is negative staining. We use an electron-dense material. In other words, you can use uranyl acetate or phosphotungstate. They scatter electrons. So you take your virus and you immerse it in one of these stains, and wherever the stain is being absorbed, it will reflect the electrons. So in these images here, they're all negatively stained. So what that means is on the left, here's adenovirus. The area around the virus is not being stained. There's nothing to take up the stain. So the electrons are passing right through to in this case, a film, and it is exposing the film or a digital detector. So think of exposed film. I don't know, any of you think of film anymore. It's the old thing, but when the electrons go through, it makes it black, it exposes it. It's a negative. The stain has absorbed to the virus particle, so it's reflecting the electrons back, so they're not causing it to be dark. So they cannot either expose the film, or nowadays we don't use film anymore, we use digital detectors to detect the electrons, but it would be the same outcome. So that's what negative stain means. And there, the next one is a hepatitis B virus, same idea. The particle absorbs some stain. Here's influenza virus. You can see a little bit of detail. Right here in the influenza virus, you can see the spike glycoproteins embedded in the membrane. And here's poliovirus, and you can see some broken virus particles that are empty, apparently. So this was great to look at viruses and see the diversity and so forth, but it doesn't have very high resolution, 50 to 75 angstrom maximum. And remember, alpha helix is about 10 angstroms in diameter. So there's no way using electron microscopy that you could see an individual polypeptide chain. All you can say is, oh, these look different. And so when the first coronaviruses were isolated in the beginning of 2020, SARS-CoV-2, people looked at them and they said, they look like coronaviruses. And that's a way that you can begin to understand what virus you have. So to get higher resolution, we have a couple of other techniques. One is X-ray crystallography. And here you take your virus and you try and make it crystallize, which makes an array of all the virus particles ordered. Very hard to do. Sometimes people spend years trying to make crystals and never get them. And then they get them and they fall apart. So this is a very, very uncertain science, but many people have gotten it to work. And when you do have your crystals, then you bombard them with an X-ray beam, and then the X-ray bounces off all of the electrons in this array, and the resulting reflections, which you can capture on a piece of film in the old days, or nowadays a computerized detector, a digital detector, you can read that image and know where each atom is in three-dimensional space. So that's X-ray crystallography. And the resolution is 2 to 3 angstroms. So now you can see individual polypeptide chains. You can see the side groups on the amino acids as well. So very high resolution. Now the latest is cryo-electron microscopy, where you don't need crystals, which is great because you eliminate all of that uncertainty. And here there were 3 individuals who developed the technology to do cryo-EM, and one of them, Joaquin Frank, is up at the Columbia Medical School. But basically, and they got the Nobel Prize for this in 2017. You take your virus solution and you freeze it. And you freeze it in a very specific way in a certain buffer. But the freezing gives it some contrast. And so that's a photograph on the top there of a frozen specimen of virus particles. And then you take electron micrographs of it. It's called cryo-EM because they're frozen. You take pictures. You take hundreds of pictures, and the assumption is in every picture of a single particle, the virus is slightly differently oriented, right? It's kind of like a CAT scan where they spin an X-ray machine around you and they can construct a three-dimensional image of you. Same thing here, except we have just on the grid, the viruses are randomly organized. And then you can take individual pictures, hundreds of individual pictures. You then do mathematical transformations to get the virus particle. You do Fourier transformations and you combine the different phases. And in the end, you can get a three-dimensional image. It's called the three-dimensional reconstruction of the original virus particles. So this is cryo-EM. There's also another version called cryo-electron tomography, where they take a single particle and they just tilt the stage in different ways to get that angular difference between the two virus particles. So they end up giving you the same thing, except with tomography, you don't have to take so many pictures. And so here's a comparison of X-ray crystallography and cryo-EM. Here's poliovirus. It was one of the first viruses to be solved, the structure to be solved by X-ray crystallography, 1985. And there it is in 2.9 angstroms on the upper left. You can see every atom in 3 dimensions. And so the X-ray crystallography gives you a file with X, Y, Z coordinates for each atom in space. And so then you can use a computer program, which you can do it on your iPhone nowadays. It's amazing. You could take the coordinates and build the virus structure. You can color it the way you want. These are colored blue, obviously, and it's depth cued. So if you consider the center of the particle, you can give it different colors as as the atoms move farther away. So the white ones are farther than the dark ones. You can get a sense for the dimensionality of the particle. Now on the right is the cryo-EM that was done not too long afterwards, and it only had at the time a resolution of 20 angstroms. You can see you cannot see individual polypeptide chains. By the way, the background is the frozen preparation that was used to make this cryo-EM. So you can see surface features. For example, you can see here, there's a star, right? 5-pointed star. We're going to learn about that in a moment. That's what we call the 5-fold axis of symmetry of the particle. And here's a 3-fold axis right here. So you can see features. But over the years, this has now improved. It's as good or better than X-ray crystallography. And you know what mostly improved? The computer algorithms that were used to interpret the data. It wasn't much in the sample preparation. It was just refining the algorithm. And now we can do 1.5, 2 angstrom resolution, and it's just so much easier than X-ray. So here are some examples to show you. This is Zika virus at 3.8 angstroms. I don't remember if this is probably cryo-EM because it's 3.8 angstroms. This is the whole particle here, part of it. This is like half of it right here. And this display is showing the alpha carbon trace only, not the side chains. And it's displayed in a ribbon format. So you can see, for example, alpha helices here and there. You can see beta strands and beta sheets, the different structural elements in the particle. And then on the upper left is a space-filling depiction where we have all the atoms shown as spheres to get an idea of how the particle actually looks. And the different virus proteins are colored differently. So you can see there is a 5-fold axis there in red with 5 subunits around it. And there are 2-fold axes, these green ones. These are 2 individual proteins, and then 3-fold as well. So you can manipulate your program to display the virus particle any way that you want, depending on what you want to see. Now, very shortly after SARS-CoV-2 was identified in January 2020, remember we talked about its identification in Wuhan initially. A month later, the cryo-EM structure of the spike protein was solved, February 2020. That's how fast you can do this. So just the spike protein, the whole particle would be very difficult to do. It has a membrane and it's fluids, so it's not going to be as easy. The protein is rigid, more or less. And so here is the particle on the left. It's enveloped and it actually has a number of proteins embedded in the membrane, but the main one is the spike, which you can see is the long one here, and it's a trimer. And there is the spike monomer with different elements that we'll talk about at different times in this course. So that's just an example of how quickly you can do these structures now by cryo-EM. And many SARS-CoV-2 protein structures have been determined by cryo-EM, and an imaging company here in New York built this model of the whole virus particle. So it has the spike structure, which you can see there. Inside is the RNA protein structure, and then the membrane they wrapped around it, and the membrane has a couple of other proteins embedded in it as well. So you can do very, very beautiful things. And there is no apparent upper limit as to the size of virus particles that we can solve structures of. This is a giant virus. We talked briefly about these in lecture 1. They're just really bigger than everything else. It's called Cafeteria Roenbergensis virus. It is a marine virus that infects Cafeteria roenbergensis, which is this unicellular flagellate, which the ocean is full of. It's one of the most common protists in the ocean. So this virus infects it. It's huge, and it's 300 nanometers in diameter. It has over 15,000 capsid proteins. I mean, this is like most of the viruses we talk about have a few hundred. This has got 15,000. And this structure was determined by Sean Chow in UTEP, University of Texas, El Paso. And he says it took him 3 million CPU hours to determine this structure. And of course, he's very proud of it. So he made a movie of it rolling on the ocean here, which if you go to that website, it has music too. Okay. All right, so let's talk about individual principles of building virus particles. Last time we talked about 7 different kinds of genomes. There are only 3 different kinds of virus particles, so we can use the subway icon again to make you remember that. I don't think I ever go on the 3 train, but it's there, 1, 2, 3. We have helical, we have icosahedral, and we have complex symmetry. These are different ways of building virus particles. I'm going to take you through each of these. Helical, icosahedral, and complex. Complex just means we have no clue how this is built. It's just really big and really complicated. Now, it turns out that Watson and Crick made a big contribution to not only DNA structure, of course, they solved the structure in 1953, but they also made an important contribution to our understanding of the structure of viruses. They noticed in looking at pictures of viruses, like here, tobacco mosaic virus on the top, polio virus on the bottom, they said most viruses are either rod-like or spherical. Of course, they didn't look at many viruses back then in the 50s, and if they had, they might have been more confused. But the ones they looked like were small, you could grow them in the lab, either rod-like or spherical. So they had this idea that since the virus genome was pretty small, and I have an exclamation mark there because now we know they're not so small, they can be bigger than some cellular genomes, they say there's not a lot of space to encode a capsid protein. So probably there's one or a few capsid proteins and they're repeated many times. So this is called genetic economy. And you know, they turned out to be right because that's how many virus particles are built, by taking one or a few proteins and repeating them over and over. And they said the rod-like viruses, the rod-shaped viruses, were built with helical symmetry, so subunits wound around a genome. And the spherical ones were built with icosahedral symmetry. So they and some structural biologists established these principles, and I want to try and explain them to you now. So we're going to use the rules of symmetry to explain icosahedral and helical symmetry. And these rules explain why virus particles will self-assemble. You can make proteins and they will assemble into a virus particle. So first rule is that every subunit, remember, subunit is a protein, single polypeptide chain. Every subunit has identical and bonding contacts with its neighbors. And I have identical in quotes because it turns out they're not always identical, but they're almost identical. And we'll see how that works in a minute. So you have a few proteins repeated over and over, so you have chemically similar surfaces, and when you repeat them over and over, you get a symmetric arrangement of the protein that's very stable in the particle. And secondly, the bonds are not covalent usually, as I told you before, and that's good for disassembly, as we said, but they're also good for assembly, because if you assemble a particle and it's wrong, you made a mistake, you can take it apart because the bonds are reversible. So symmetry rules, these are the two rules of symmetry. Now these are useful for us, which means because if you take viral proteins and produce them on their own, in many cases they will assemble into virus particles. We call them virus-like particles, VLPs, because there's no genome in them. So to be a virus, they would have to have a genome. But you can just make the protein and you get these particles. And we have at least 2 vaccines that are made this way. The hepatitis B virus vaccine and the human papillomavirus vaccines are virus-like particles made in yeast. You put the gene for the capsid protein in yeast. The capsid proteins are made. Here's the individual capsid protein here on the left. They assemble into a pentamer, 5 copies of the capsid protein, and then the pentamer assembles into an empty particle. It's all the information for making the particle is built into that protein. So we can make vaccines of various sorts, and when we talk about them, we'll discuss how really good these vaccines are. All right, now back to the symmetry. First, helical symmetry. Here we basically have a single protein, and for tobacco mosaic virus, which is shown here, it's called the coat protein, which is interacting with each other and with the viral RNA. And so here you can see the schematic on the upper right. The RNA is shown in green, and it's coiled in a helix-like structure. And these coat proteins are bound to it. And you can see there are interactions among the coat proteins and also with the RNA. So you end up with a very stable structure, which is shown here. And for tobacco mosaic virus, that's the virus particle. It's just a protein repeated many times bound to the RNA. Lower right is the electron micrograph of TMV, tobacco mosaic virus, rod-like particles with helical structure. You can have many other viruses with helical symmetry. There are viruses of animals with this kind of symmetry. Here's Sendai virus, which is a paramyxovirus related to measles virus. It has a longer structure. It has a coat protein, which we now call nucleocapsid protein, NP, bound to RNA. As you can see, it's much longer, 1,000 nanometers in length. The interesting thing here is that for animal viruses with helical symmetry, they always have an envelope, as opposed to TMV, tobacco mosaic virus, which isn't have an envelope. Apparently, they can go from plant to plant like that. Animal viruses have to be enveloped, and here's an electron micrograph of a paramyxovirus like Sendai virus, where one of the particles is broken and the nucleocapsid is spilling out. That's called a nucleocapsid. This structure up here is called a nucleocapsid because it's a substructure, has an envelope around it. So once it has an envelope, it becomes a substructure, and that is called a nucleocapsid. The tobacco mosaic virus is not a nucleocapsid because it's just a naked helical nucleoprotein complex. Just a little detailed look at how this works for an animal virus. So here's vesicular stomatitis virus related to rabies virus. People work on this because it's safer to work on. It's a virus that infects cattle, and it is bullet-shaped. It has a helical nucleocapsid, which is encased in an envelope. And it's built the same way. You have a nucleocapsid protein that is bound to the RNA and interacts with each other. So here in panel A, there's a structure of the nucleocapsid protein, multiple subunits, joined to a piece of RNA, which you can see in green. And they make a ring, which would be part of this helical structure. And then here's a single nucleocapsid protein. You can see there's a groove around the middle of it where the RNA fits in. And so that's how this protein interacts with the RNA. and forms this helical structure. Here's a cryo-EM of the VSV helical structure. On the left are the EMs that were used to generate it, and you can see that the nucleocapsid RNA structure coils within this particle. It's got an envelope around it, and actually ends up in a bullet shape like that. So there are a number of enveloped RNA viruses that have negative stranded genomes and helical capsids. They're shown here. We're going to talk about some of them. Measles virus, for example, paramyxoviruses, rabies, influenza virus, Ebola viruses. They all have nucleocapsids, which are the nucleic acid proteins assembly that are packaged within the particle. So you can see for each of these, there's a distinct substructure of the RNA complexed with proteins that makes it a nucleocapsid. So here is a breakdown of the different ways you can make nucleocapsids. So wherever there's a nucleocapsid, it's labeled. Wherever there's no nucleocapsid, it's not labeled. Look it up at the top here. We have a virus that looks like tobacco mosaic virus. There's no nucleocapsid. There's no substructure. The RNA protein complex is the whole virus itself. On the right is a icosahedral virus with a genome in it. Could be RNA or DNA. It's not a nucleocapsid. There's no substructure. But if you put membranes around these two viruses, now you would have a nucleocapsid. This helical structure would be a nucleocapsid. This capsid would now be called a nucleocapsid. The idea is that it's kind of like the nucleus of a cell, right? It's inside the cell. On the bottom, here we have envelope viruses, but the genomes in them are not complexed with any protein. That's one of the requirements to have a nucleocapsid. So there's no nucleocapsid structure there. If you had proteins on them, they would be nucleocapsids. SARS-CoV-2 is in the upper right. It has a nucleocapsid because the RNA is complexed with proteins. Those are those spheres there bound to the RNA. And here is a non-enveloped virus that has a nucleocapsid. This is a Mimi virus, a giant virus, where it's icosahedral symmetry, but inside is a helical nucleocapsid, DNA bound to protein. So remember, the substructure is what makes it a nucleic acid protein substructure within the virus particle. That makes it a nucleocapsid. Here's SARS-CoV-2 as an example of enveloped plus strand viruses with helical capsids. There are lots of coronaviruses that are like that besides SARS-CoV-2. There was the original SARS-CoV, MERS coronavirus, and many, many others. They're envelope viruses with RNA protein complexes in the interior, and they are nucleocapsids because they are protein plus RNA equals nucleocapsid. And here are some electron micrographs of coronaviruses. This one on the top right, one of the first, and the people who took it said, it kind of looks like the solar corona. So they called it coronaviruses. Did you know at the beginning of the pandemic which New York City neighborhood had the highest rate of COVID positivity?
Was it Corona?
Corona Queens, yes.
Wow.
You couldn't make it up, right? So the solar corona. And then on the bottom is SARS-CoV-2, where they could see similar spikes. So they said, aha, this must be a coronavirus, which you then confirmed with nucleotide sequencing. So there are both DNA and RNA viruses with helical symmetry. Just so that you know, you don't have to memorize any of this. Both nucleic acids can have helical symmetry. We looked at tobacco mosaic virus, which would be a plus RNA, but there are also minus RNA viruses that are rod-like or flexuous. That means that they are not rod-like, they're flexible. There are double-stranded DNA viruses that are flexible. There are DNA viruses that are rods. And there are also single-stranded DNA viruses that are flexible. So the helical symmetry can accommodate a lot. So in my studio, I have all these magnets, these tiny, very powerful magnets, buckyballs, which for a while you couldn't buy because kids were swallowing them, right? And then they came back on the market. I don't know why. And so you can build all kinds of virus particles. So this is a helical virus built with buckyballs, and you can unwind it and see exactly how a helix works. So I think they're very cool. And I'll show you how you can build icosahedral particles with those as well. All right, we have to do another quiz here. Number 2. Which of the following describes virus symmetry and self-assembly? The bonding contacts of subunits are usually covalent. The bonding contacts of subunits are usually non-covalent. Each subunit has different bonding contacts with its neighbors. Self-assembly of virus particles does not occur, none of the above. So which describes virus symmetry and self-assembly? 98% got the right answer. The bonding contacts are usually non-covalent, right? They're not covalent. That's number 1, no. And nobody took the others, good. They're not usually covalent, they're usually non-covalent. Icosahedral symmetry. So we have to make a round virus now. How do you make a round virus from proteins that are irregular? So here's a viral capsid protein. It's a poliovirus capsid protein. Poliovirus makes spherical particles. And how do you make, that capsid protein is irregular. So how do you make a spherical particle? So a number of, Watson and Crick and a number of crystallographers made a hypothesis, and they based it on 2 bits of evidence. First, if you look at round viruses, they have multiples of proteins, usually multiples of 60. 60, 180, 240, 960. There are exceptions, but they noticed that this was common. And secondly, even though spherical viruses can be tiny or very large, the capsid proteins are more or less the same. They're about 20 to 60 kilodaltons in size. And so Watson and Crick said this is consistent with building a spherical virus with what we call icosahedral symmetry. An icosahedron, as we see here, is a geometric solid, one of the Platonic solids. It is a solid with 20 faces. Each face is an equilateral triangle, so there are 20 of those. And when you build a solid like that, you get axes of symmetry. You get 5-fold, 3-fold, and 2-fold axes of symmetry. All those mean is that there are 5 subunits around, I don't want to use subunit, that's a different word. There are 5 structures around the 5-fold axis, so for each axis. So for 5-fold axis, you can see there are 5 triangles around it. For the 3-fold axis, there are 3 sides of a triangle around it, and for a 2-fold, there are 2 sides of a triangle. You can see that actually better right here. The 5-fold here in green, the 3-fold, and the 2-fold axis. So it turns out that an icosahedron is favored because you can make a closed shell with very good stability with just 60 subunits. So 60 proteins, 60 of the same protein, you can make an icosahedral particle, and it's very stable. So none of the other solids, and there are other Platonic solids as well, none of them will do that. And so probably this is why viruses have evolved to have this kind of symmetry. So the simplest icosahedral capsids have 60 structural units made of 1 protein. So remember, the subunit is the protein. The structural unit is made up of 1 or more proteins. So for these viruses, the subunit and the structural unit are the same. 1 subunit per structural unit. So there's the structural unit at the top. That's the protein, but it's also the subunit. Sorry, this is the structural unit, but it's made up of a single subunit or protein. And these are repeated 5 times around the 5-fold axis and then individually to make the virus particle. So 60 units made of 1 protein. So we have 60 proteins in this virus particle. And amazingly, all those proteins interact identically with their neighbors. So this was the whole idea that arose originally from Watson and Crick, was that all the interactions of proteins in these particles would be identical. We know that they're not anymore, but they are for this instance, the viruses with 60 units made of 1 protein. But even though I'm showing these diagrams as icosahedral, the particles are not icosahedral, they're spherical, as you'll see in pictures. Icosahedral symmetry simply refers to the arrangement of the proteins in the particle. It doesn't mean that the particles look angular like this. So here's an example of a simple viral capsid, adeno-associated virus, which is a parvovirus. Again, these are viruses that can infect you or your pets. They're also used for gene therapy applications, which we'll talk about later. But they're made up of a single protein, 60 copies of a single protein. There's the protein right there, and 60 of those are arranged in icosahedral symmetry in a way that I just told you to form the capsid. And you get 5-fold axes of symmetry there in blue, 1, 2, 3, 4, 5 around it, and 3-fold and 2-fold axes as well. That's the simplest structure that we can do. Now, how do you get a bigger virus? Because lots of viruses are bigger. We do not make bigger capsid proteins. We make more subunits per particle. We add subunits. So here is a bigger virus with 180 identical protein subunits. The previous one had 60, so it was small. Now we're getting bigger. We add more subunits. And now, when you add more subunits, the rules change. The interactions among the proteins are no longer identical. They can't be in order to pack them in. And the clearest way to illustrate that is to say that when you put more than 60 subunits, and this is the next size that you can do, you don't do anything in between, you now have different modes of packing of the proteins. And so, whereas in the previous virus, we had only pentamers, associations of 5 proteins. Here we have pentamers and hexamers. So here on this particle, you can see the proteins on this particle are shown as commas. So each comma is a protein. So here's 1, 2, 3, 4, 5 protein subunits. That's a 5-fold axis of symmetry there, shown in green. But you can see there are also, that's a pentamer, there are also hexamers in purple. 1, 2, 3, 4, 5, 6 protein subunits around a 6-fold axis of symmetry. That's the red triangle there. All right, so here now we no longer have identical interactions because some of the proteins are in a 5-fold environment and some of them are in a 6-fold. So we call this quasiequivalent because they're similar. The interactions among the proteins are similar but not identical. That was the additional definition we had to make to explain these bigger particles. So quasiequivalent simply means when a capsid has more than 60 copies, each occupies a quasiequivalent position. It's similar but not identical. The binding properties are similar, so they're non-covalent as always. But they're not identical because you have 5-fold associated structures and 6-fold associated structures. So now I've been showing on these slides a T number, T equals 1, T equals 3. That's called the triangulation number. And what it is, in the simplest terms, it's a pretty complex mathematical description that I don't want to give you, but in the simplest of terms, the T number is the number of subunits that make up the structural unit. So remember, the subunit is the protein chain. It's the number of protein chains that make up the structural unit. So for the T equals 1 that we talked about, the structural unit and the subunit are the same. So there's one subunit for structural unit. That's T equals 1. And you can use the T number to calculate the total number of subunits in a particle. It's T times 60. The next size virus is a T equals 3, where there are 3 subunits per structural unit. And so that has 180 subunits. And then you can have a T equals 4 and a T equals 13. You can get bigger and bigger. Anything bigger than T equals 1 now has both a 5-fold and a 6-fold axis of symmetry, as I just showed you. So this is how you make bigger particles, by simply adding more subunits. So I made a video using buckyballs. You can go look at it. You can make icosahedral particles, and it really illustrates the difference. So there is a T equals 1 particle on the left, and it's made completely of pentamers. So here are 5 magnets in a pentamer, and you can use these to build that T equals 1 particle, and you get 60 subunits in it. when you build a T equals 3, it will not work with all pentamers. You have to use hexamers. So you can see the red pentamers, the blue hexamers. And so you have pentamers at each 5-fold axis of symmetry, and then everything else is a hexamer. There's just no way it works by using all pentamers. So that's a nice illustration of how you can do this. I was visiting some university, was it Indiana? And I went to talk to a chemistry professor, and he had these on his shelf. And I said, I didn't know you could build those. So he showed me how to build them, and now I made this video. I just think it's great. It really illustrates how this works. All right, so here's an example of a T equals 3 virus, poliovirus. It's an icosahedral capsid made up of 3 subunits per structural unit, so it's T equals 3. Now, it turns out that those 3 proteins are different. They're called VP1, 2, and 3. So this is actually pseudo T equals 3 because to be T equals 3, they have to be identical. But they're similar enough that it's still a T equals 3. 3 subunits per structural unit, T equals 3, 180 total subunits. And that's the particle here with its 5-fold and 6-fold axes of symmetry because it's a T equals 3 particle. We can get bigger particles. This is a polyomavirus, SV40, that we're going to talk a lot about in this course. It's made up of 72 pentamers of VP1. So here is a pentamer here, 1, 2, 3, 4, 5. But some of these have 5 neighbors, and some of them have 6 neighbors. And so this is a big particle, so it has pentamers and hexamers. So here is a pentamer with 5 neighbors, 1, 2, 3, 4, 5. And this pentamer has 1, 2, 3, 4, 5, 6 neighbors. So pentamers and hexamers. So this is a bigger particle, and therefore you need to have both pentamers and hexamers. This is a DNA virus, and I wanted to show you how this is built because it's really cool how this is held together. So this is VP1. So each of these proteins, the yellow, the green, the blue, etc., each of those is a VP1 monomer, and VP1 is shown on the left here. This is a pentamer. You see there's a protein, there's the N-terminus sticking out from each subunit, right? Here's the N-terminus of each VP1. Those are going to grab the next pentamer. I'm going to show you that on the next slide. So now everything is blue except the central pentamer. That's surrounded by 5. And look at the N-terminal extensions are wrapping into the next pentamer because you can see the yellow and the blue in there and the green and the blue. So they're like hooks holding them together. So that gives the particle stability. So it's a very simple structural solution there. Yes?
So I'm a little confused because you keep saying that the subunits are identical, or is it the structural units or the subunits are identical, and there's 60 of them, but that there are different subunits making up the structural units?
So the subunits in the simplest viruses are identical. So here, they're all VP1. They're all the same protein. They're arranged in structural units. Here, the structural unit is the pentamer. For polio, it was a trimer. It's a different structural units. As we get bigger, we're going to see there are other proteins involved, but the basic icosahedral structure will always have one or a few proteins repeated over and over to make the structural unit. So we have, say, 1 or 3 subunits to make up structural units.
So when you say icosahedral, that is can there be different subunits with it, like different protein subunits?
There can be, like for polio, there are 3 different protein subunits. Most of the time, they're identical, though. That's why we call polio pseudo T equals 3. There are other T equals 3 viruses where the 3 subunits of the structural unit are exactly the same protein. Okay, so most of the time, they're the same protein. All right, next question. Which of the following are characteristics of icosahedral symmetry in viral capsids? Produces a solid with 20 faces, each an equilateral triangle. Allows formation of a closed shell with 60 identical subunits, 5 3-fold and 2-fold axes of symmetry. The T number describes the number of protein subunits per structural unit, or all of the above. All right, let's see what we did. 92% got the right answer, all of the above. Yeah, these are all characteristics of icosahedral symmetry. That's the solid with 20 faces, closed shell with 60 subunits, 5, 3, and twofold axes of symmetry, and the number of protein subunits per structural unit. All those are correct. Now you can make big capsids, as we said, by adding more subunits, but then you end up adding other proteins that are not particularly symmetrical to have other functions. So here, adenovirus is a big capsid, 150 nanometers. Its T equals 25, and it has proteins with specialized roles. So for example, here is the icosahedral capsid, and you can see the green spikes. They are coming out of each 5-fold axis of symmetry. And this is the structure of the spike all the way on the right. It's a trimer of a single protein, which is then inserted at the 5-fold axis of symmetry. So that's a specialized function. It actually attaches to the cell receptor. There are also other proteins in this capsid. It turns out that when you make a big particle and you have pentamers and hexamers, it's not very stable, because if they were all pentamers, like in a T equals 1, it would be really stable because you have exactly the same interactions among all the proteins. But once you make bigger particles, they're less stable. So additional proteins are stuck in here to make the particle more stable. You can see some of these are these brown proteins here, which are stuck in, protein 9, they're stuck in between the subunits. We call them glue because we think they stabilize the structure. So the point here is that as you make bigger and bigger particles, you can make specialized structures. Some viruses actually have 2 icosahedral protein layers. These are reoviruses that are double-stranded RNA viruses. They have 2 shells with different T numbers. They have an inner shell, which is a T equals 2, and they have an outer shell, which is a T equals 13. And here is a schematic of that. The inner shell is in light tan, and the outer shell is in purple. This has a very specific function in the reproduction cycle, as you'll see later, because the outer shell comes off when the virus infects the cell, but the inner shell stays on. And why that happens, we will talk about. Bacteriophages are kind of hybrids, or they can be hybrids. Not all phages look like this, but these are the tailed bacteriophages, where you have an icosahedral head, where the DNA is inside, and you have a helical tail attached to it. So that tail is built with helical symmetry, even though there's no nucleic acid in it, it's just a protein tail. And then you can have tail fibers, as you can see here. They're not always present, but they're a different structure, obviously. And then at the bottom, there's a base plate, and that's where the phage will attach to the bacterium, and it's wider than the tail, and has a feature that allows piercing of the bacterial membrane. So this is another version of the bacteriophage structure. For many phages, when the phage sits down on the bacterial surface, the tail contracts and inserts a spike into the bacterial membrane to make a hole, and then the DNA comes out of the tail, out of the head through the tail into the bacterium. The DNA is packaged at hundreds of pounds per square inch in these capsids. So as soon as there's a hole in the bacterial membrane, the DNA shoots out. But the spike is an interesting protein. Here's a base plate on the left, and you can see the red spike there. And that is actually a trimer of a single protein in red, blue, and green, you can see here, which is made up of largely beta strands that get smaller and smaller and taper to make the point that is the spike. So that's a really cool way to make a spike that has to go through a membrane. And there's, at the very tip, there's an iron molecule that's bound to all 3 chains. You can see that single iron is bound to all 3 chains and helps to stabilize the spikes. So again, specialized structures. Here's another example, herpes viruses, where we have a nucleocapsid. These are envelope viruses with an icosahedral capsid containing DNA. So it's a nucleocapsid, as you can see here. And the structure of that nucleocapsid alone is shown on the right here. It is composed, it's a big particle, so it has pentamers and hexamers. as usual, but it has a specialized structure called the portal. There is one portal per particle, and you can see, this is the portal right here. It's basically a hole through which DNA goes in when the particle is built, and through which DNA comes out when these viruses infect cells. And there's just one per particle. And you can see here's an electron micrograph where they are using antibodies to one of the portal proteins to stain it. And you can see one portal protein there and one portal protein there. So it's at one of the five-fold vertices, and it's needed for entry and exit. And I have actually keychains that people have given me over the years. I have dozens of them. And this is a herpes virus keychain. So this is the herpes virus. You can see the envelope with the spikes. And then it opens up, and you can see the nucleocapsid inside. And there's a portal. They actually put the portal there. And then you can open the nucleocapsid and see the DNA inside of it. This is the ultimate geeky keychain, but it's, for us, now it makes sense, the structure for you, right? These capsids can have membranes on them, as I've said previously. The membrane's always from the host cell, because the viruses do not have the ability to make lipids. And typically these membranes are acquired as the virus moves through the membrane by a process we call budding. And that's illustrated here. You can see this is happening at the plasma membrane. The viral proteins are being assembled under the plasma membrane, and then a bud forms, pinches off, and you have a virus particle. And this doesn't have to be the plasma membrane. It could be the ER, it could be the nuclear membrane, it could be the Golgi. But for every virus, it's always the same. It doesn't vary. And of course, if you have a protein nucleic acid complex within the envelope, it's a nucleocapsid. That can be helical or icosahedral, as we've already talked about. Now, in these membranes are glycoproteins. These envelope viruses have glycoproteins embedded in the membrane. We call them spikes, the famous spike protein of SARS-CoV-2. This is influenza virus, which has two different spike proteins in its membrane. They are very important. They're needed for cell attachment. They are antigenic sites, so immune responses directed against the spike. We'll talk about that later. They're involved in assembly of the particle. And they're typically oligomers, like trimers. And they look in the electron micrograph like spikes. So this is one of the first EMs of influenza virus taken. And you can see the very distinct spike-like structure on the surface of the particle. And that's why we call them spikes to this day. Some of these spike proteins are perpendicular to the membrane, like the influenza virus hemagglutinin shown here. But they don't have to be perpendicular. Some others are parallel to the cell membrane. These flaviviruses, the spikes are actually flat on the surface of the particle, yet they can still serve their function as attachment proteins. And you'll notice that for these viruses, these flaviviruses, the spike proteins are actually icosahedrally ordered, right? You see 5, 3, and 2-fold axes of symmetry. The particle, there's no particle underneath it. There's just the membrane and the nucleic acid, as you can see here. Yet these proteins have the ability to arrange themselves in icosahedral order. So we have two kinds of enveloped viruses that we see. We see envelope viruses with helical nucleocapsids, influenza, Ebola virus. We have envelope viruses with icosahedral nucleocapsids, herpes virus, toga virus. So the nucleocapsid can be either type. And finally, complex particles don't have any rules of symmetry. They're made of different structures, so pox viruses, Pandora viruses, pithoviruses, they typically have membranes. They have a large core containing DNA, like you see in the pox viruses. But you see Pandora virus, the DNA is extended. It's different in pithoviruses. So these are made up of many, many hundreds of proteins in no particular symmetrical order. That's why we call them complex particles. And just remember that there are other things in virus particles as well besides these structural proteins that make up the particle. There are often enzymes of various sorts. We'll talk a lot about reverse transcriptase. And there are other proteins that have to do with early events in infections. They don't have a structural role, but we consider them to be components of the virus particle. All right, now you know how to build a virus. So next time, let's talk about how to get into a cell and begin the infection.